With chemicals, we can classify chemicals or lump them into groups based on just knowing a couple of key things. Just by knowing the molecular structure of it tells you something about where it's going to go in the environment and where it might end up, or what other chemical it might be similar to or behave similar to because of the way that it's shaped, or the way that it looks, or the way that it interacts. Okay? With nanoparticles, this is much more complex. For chemicals, we don't really have shape. Now, with nanoparticles, we do. We actually have a physical shape. Okay? But the thing is, we don't know which ones of these things are important or predictive of where they might go in the environment and what they might do. And so that's where a lot of studies in my lab and at OSU and within Oregon are focused now, is trying to figure out to tweak each of these and see, well, how does that impact now the toxicity of the material? So we can start building the database that we need to understand these relationships. Okay? Not only do we not really know what to measure, what's important to measure with these nanomaterials, but we don't really know how to measure the dose of these materials. When you look at chemicals, every chemical that has been tested is in milligrams per kilogram body weight. The mass of the material or the chemical compared to the mass of the organism that's being exposed. Okay? With nanomaterials, mass is not all that easy to get at. So since in toxicology the dose makes the poison, it's really, really important to figure out how to measure or what to measure as far as the dose of these materials goes, so that people are being consistent about it. It's easy in the chemical world because it's the standard protocol to measure things in milligrams per kilogram body weight. For nanomaterials, that's not the case. And so when we look at mass, do we look, here's little Joe nanoparticle here, okay? He's got a little core, and then he's got some sort of stabilizing shell, and then some chemistry on the surface, okay? Those are the little decorations, the ligands on the surface, okay? So when you're talking about the mass, are you just looking at the mass of the core of the atoms that are there? Or are you looking at the total mass of this whole group? Because that's going to be very, very different. Are you looking at the number of atoms that are in a cluster or a particle? Or are you looking at, is it the number of particles that matter? Okay? Is it the surface groups, okay, the number of ligands that might be attached, or the type of ligand that might be attached? Or some studies have now shown that it's actually the surface area that's most important to know. So even just getting at something as simple as measuring the dose that you're exposing an organism to is inordinately complex compared to what we know in what we've relied on in the chemistry field. So we don't really know which dose metrics are appropriate. And then to top this off, we have this problem of experimental design. Okay? So for biological test systems, you can choose to look at something in a whole animal system, okay? You can look at it in cell culture, okay? Depending on the species that you pick, the age of the animal, the sex of the animal, what particular cell line you selected, those all impact what your final results are, okay? So having a standard, model organism is critical. And on top of that, we have the other part of that, which is the exposure scenario. How did you expose this organism you selected to the nanomaterials? And here, the dose or the concentration makes a difference. Mm-hmm.
Yeah, how do people manipulate this stuff? I mean, somebody's working with the nanomaterials. What are they actually doing?
Ooh, that really depends on who you are. Can you give me an example? Like, well, I mean, somebody in the lab?
Yeah, somebody in the lab. They're not pouring it from vial to vial. I mean, I don't know how you actually manipulate this stuff.
It's very challenging, and so usually you make your original solutions much more concentrated than the solutions that you'll use to expose, because otherwise you just can't weigh it out. And then a lot of the, like our balance in our lab is electrostatically lined with anti-static screening. And then we also go in with a little brush to get rid of any more static that's in there. Because some of these materials, as soon as you open up the vial, they just go and that's not good, because inhalation exposure is not wanted.
How do you protect the workers?
So we have, in our lab, we treat all of the nanomaterials that we test as hazardous materials. So we use all of the precautions that you would use if you had a known toxic agent in the lab. So we use all those same protocols, but then we double glove for the materials that we know cross the barrier very easily, or for materials that we know are extremely hazardous. Double glove and use extra precautions.
It would seem to me that these could penetrate gloves, masks.
Well, and the National Institute of Occupational Safety and Health has done assays of that, where they found actually some of the ventilation systems that you use are really, really good at taking out those little particles. Okay, so they'll take them and they'll agglomerate basically on a static charged membrane. But the gloves that they've tested have pores that are larger than the size of a lot of the nanoparticles. Mm-hmm.
You talk about dilution. Now, to me, dilution is just add water, but I wonder if the water is probably out of all conceivable scale to this. I mean, how do you actually dilute it?
We do it, well, it's more considered a dispersion, like when you're talking about the particles in water. So if you think of it more as like a colloidal dispersion, then you can kind of wrap your head around it. So dilution, and most of the materials that we work with in our lab are in aqueous solution. They're already in water, or we treat them so that they'll solubilize into water.
Mm-hmm. There probably haven't yet been any human studies done, but there are probably protocols going on in which humans are involved. So I was wondering if there are any human subjects research committees that are involved in any of this stuff.
Well, anybody in here purchased some of that silver spray for in your mouth that gives you fresh breath? There's colloidal silver that you can buy over the internet, and if you go to some of those websites, you will be amazed what nano silver can do for you. I mean, it can cure every ailment you've ever had. You'll never be healthy. You'll probably live forever. So there are human subjects that are out there that have been taking those types of nanoparticles for quite a while now.
But are there human subjects committees that have been reviewing any of these?
No, because we're not even there yet. We're not even close. But all of the regulatory agencies are frantically trying to get ahead of this so that they can base regulation on some sort of, scientifically valid data, which they're having a really hard time even generating. Mm-hmm.
Don't you also have the problem of having to study the degradation products as well as the main product?
Yep, and that's a big one with silver nanoparticles because they do dissolve. It takes them a very long time to dissolve, but they do dissolve, and they've shown with some of the zinc oxide nanoparticles that dissolution rates completely drive the toxicity of the material. So there's that factor as well. So that's another thing, is that chemicals, for the most part, may be modified a little bit as they move through the environment, and they may be tweaked a little bit, but nanoparticles are truly dynamic. So they take on, whole surface chemistry. They pull in all the organics around them and it binds to them, and it changes depending on the environment that it's in. So they're very dynamic. So that adds another level of complexity to the materials. Okay, so dose and concentration, obviously an impact. The timing, like from the time that you're born to death, where you are exposed along that continuum makes a difference as to what the impact might be. The duration, were you exposed for over your entire lifetime, or were you exposed a couple different times at small periods during your lifetime? Those all make an impact on the results that you're going to get. So it's very, very complicated. Then lastly is the route of exposure. So how you're exposed to it, whether you're exposed to it through the skin here, or inhalation by inhaling it, injection, which a lot of the nanomedicines are going to be injected, or oral exposure. So depending on how you get this in you, makes an impact on what it's going to do within the organism. So then how do we deal with all this? Okay, all of this complexity, it makes my head spin. And when I first started into this, I thought there's no way I'm ever going to be able to organize even just a data set, let alone organize all the concepts to make actual data turn into some sort of information and knowledge. And I realized at that point that informatics and computational tools were my friend, and I needed to start moving in that direction, and that's part of what my lab does now.
Can we go back one slide?
Uh-huh.
Is that you?
Yes.
It is.
Yes, that's me, and that's me.
Oh.
And this is not my child. I have 3 of them here, but this was on the internet, and she was really cute. So. Okay, so what I propose is that instead of looking at, okay, we did this cytotoxicity test and we had a negative response, and we did this test, and doing all these canned tests that we typically do with chemicals, is really stepping back and taking a weight of the evidence approach, where you think about things in a broader sense as to the impacts that might actually occur. And if you just take it in its very broadest sense, any risk that you're going to have from these materials are a function of how toxic they are, how inherently hazardous they are, and the exposure. Okay, how long you were exposed to them, if you were exposed to them. If they're in a tennis racket, your exposure is going to be next to nothing. Okay, if it's the colloidal silver that you're spraying in your mouth every day, your exposure is going to be really, really high. And so we take more of a broad approach using all of the animal and the cell culture studies and data being collected in various different laboratories, national labs, and academic labs from around the world, and try and use those, pulling everything together into some sort of framework where we can start understanding nanomaterial-specific environmental health and safety considerations. Now this, where you're using weight of the evidence and pulling all of this data together, has not really been done for chemicals. I don't know if you don't know that. But that's not really been done for chemicals. They literally have the tests that you do, and those are the tests that chemical companies are required to do. But taking a weight of the evidence approach and stepping back and going, okay, maybe the model systems we've been using are, they're not the fastest, okay? They may not even be the most accurate. So maybe we should step back and reevaluate how we're even assessing these materials, okay? And then where this plays in very nicely is that in the past, these nanotechnology applications have, just gone forward. They've gone forward and they've got their final product and they've got their whole nano manufacturing down, and then they say, here's the end product. Toxicologist, can you test it for me? Can you tell me what a life cycle assessment might look like for this particular material? With nano, we're trying to move to a point where these pioneering applications are actually done at the same time that we're evaluating them. We're evaluating the hazards of these materials and the hazards associated with the process, the processing of these together, hand in hand, while they're trying to optimize performance, trying to minimize hazard at the same time. It makes sense. And by building this strong bridge between the applications and implications research, we can actually start to think forward and anticipate any potential problems, and then try and get around those with some solutions.